Welcome, everyone. It's our great pleasure to welcome you to another Meet the Drug Developer webinar. I'm Andy Roberser from the Myotonic Dystrophy Foundation, and today it's our great pleasure to welcome Juvena Therapeutics to provide us an update about their exciting news and development. The myotonic dystrophy, for us at the Myotonic Dystrophy Foundation, our vision is, of course, that we see a world with treatments and a cure for myotonic dystrophy. The mission of the foundation is community, care, and a cure. So we connect and support the myotonic dystrophy community. We provide resources and advocate for care, and we accelerate research towards treatments and a cure. As you hopefully all know, our website contains many resources and support. So we have toolkits and publications. We have support programs. We have our calendar that highlights all important events. And we host the Digital Academy, where you find hopefully later today or sometime early next week, a recording of today's webinar. Also very, very exciting, and many of you have heard and seen this. We are holding in 2025 an annual conference. This conference will take place from May 2nd to May 4th in Indianapolis. Great venue, working on a great program, and looking forward to seeing you all there. July is also Myotonic Dystrophy in Motion Awareness Month. We have two more events. Week 3 is the natural world, the benefits of nature and breathwork. And then the activities conclude in week 4 with Let's Keep It Going and a virtual Zumba class. So register, and again, this is on our website. On our website is also the sign-up mechanism for the Meet the DM Drug Developer webinar series, where we hear today, of course, from Juvena Therapeutics, in August from PepGen, and we already have scheduled in October Arthex Biotech. Drug development and a robust drug development pipeline is really the best indicator of hope and progress. And we in the DM community, we are really fortunate to have a robust pipeline. So as you can see here, this is also on our website, we have 20 plus programs that have significant myotonic dystrophy clinical development programs, and 9 of them are actually in the clinical testing phase, and many are approaching this stage, and this is really, really exciting. So clinical trial activities, they really fall into 4 broad areas of drug discovery, and they follow the essential dogma of biology. And so going from left to right, we start with the DNA, which of course harbors the genes, the blueprint instructions to make out the functional components, the proteins. And through a process of transcription, we make RNA first. And in DM, the expanded repeat makes it from the DNA to the RNA. Then we have a step, instead of normal folding, we have misfolding and these hairpin loop formations, and they act as ginormous traps, and they bind and recruit important cellular proteins so they cannot act and perform their downstream functions. And the result is global cellular dysfunction and ultimately disease manifestation. So the next. The activities, drug discovery activities, fall into this first area where we have inhibition of transcription and gene editing and tuning. These activities are today less mature than, for example, the next one. where we target the inactivation of the toxic RNA. Then the next one, we compensate for these trapped proteins, and in the last category, we broadly compensate for cellular dysfunction. So next. So that's where Juvena Therapeutics focuses their efforts in. So today we have a very exciting panel. We hear first from Dr. Hanadie Yousef. She's the CEO and co-founder of Juvena Therapeutics. She received her bachelor's degree from Carnegie Mellon and a PhD from UC Berkeley. She then completed postdoc training at Stanford. And as you can see here, she has received numerous very prestigious awards in entrepreneurship, innovation, and some of them, it's a long list, and maybe you can see award Woman Who Leads in Life Science, to all the way to emerging pharma leaders, a prestigious recognition in the community. Then we hear from Dr. Jeremy O'Connell. He's the Chief Scientific Officer and also a co-founder. He earned his PhD from the University of Texas in Austin and subsequently completed postdoc fellowship at Harvard University. So he has deep domain expertise in proteomics and system biology. And at Juvena, he oversees the research, strategy, preclinical science and manufacturing, as well as IP efforts. Dr. Barney King, he's the Chief Medical Officer. He's next. He received his undergrad degree from the University of Notre Dame and then his medical degree from the Ohio State University. He's board certified in cardiology and internal medicine, and he also holds an MBA from Wharton. He has extensive experience managing businesses and development operations in the pharmaceutical, medical device, and biotech industry, as well as in CROs. And then we also hear from Dr. John Day. And we all know Dr. John Day as a community member, board member for MDF. However, he's not going to talk to us today in this capacity. He will talk to us as an advisor for Juvena. So just as a reminder, Dr. Day received his MD from the University of Minnesota and a PhD in neuroscience from Albert Einstein College of Medicine. Then he completed neurology and neuromuscular training at UCSF, and then he joined the University of Minnesota as a professor of neurology and pediatrics and genetics. And there he actually founded the Wellstone Muscular Dystrophy Center. In 2011, he switched to Stanford University, where he established the comprehensive division of neuromuscular medicine. He has, of course, investigated the genetic causes and multisystemic effect of a whole bunch of neuromuscular diseases, and he brings more than 30 years of experience designing and directing clinical trials. So, a bit of housekeeping before we start. So please ask questions. Please ask them live. We already have compiled a long list of questions, and after the presentation from Juvena, we are actually looking forward to a quite fluid conversation. So, without further ado, let's welcome Juvena and their presentation.
Great. Well, good day, everyone. It's such an honor and pleasure to be here to have the opportunity to really provide an overview of our lead program, our candidate JUV161, which is a muscle regenerating fusion protein that we're developing for myotonic dystrophy. Next slide, please. Really quickly, just to note, some of the statements in this presentation are forward-looking and should be interpreted as such. Next. So at Juvena Therapeutics, we are a biopharma company that is dedicated to mapping the therapeutic potential of proteins secreted by stem cells and then translating them into engineered biologics or protein-based therapeutics that can restore tissue function in order to treat muscle, and metabolic diseases. We're doing this through a fully integrated, artificial intelligence-enabled drug discovery and development platform. We have built this platform at Juvena over the past 6.5 years, and it is really based on over a decade of research, really underlying changes in protein signaling and mechanisms underlying the biology of aging and tissue degeneration that we accrued over a decade through our work at Stanford, at Berkeley, at Harvard, and other top institutions. Now at Juvena, it is ultimately our mission to regenerate and repair cells in order to restore tissue function to rejuvenate lives. And we're doing this by taking advantage and really unraveling the therapeutic potential of secreted proteins. Secreted proteins are the signaling proteins and in many ways the worker bees of our bodies. They're proteins that are circulating in blood or localized in tissue microenvironments. And when they bind receptors on cells in our body, they induce all sorts of reactions, from modulating the immune system and activating immune response to infection, to inducing repair processes, to really signaling to stem cells in our body to activate and repair and regenerate tissue. And so you can think of many therapeutics today that have saved hundreds of millions of lives that are based on natural occurring signaling proteins. Think of drugs like, for example, and proteins like insulin, which is a protein that regulates blood sugar levels and instructs cells throughout the body to uptake glucose from the blood. Or think of drugs like human growth hormone, which is a protein that that naturally instructs cells to build muscles and bones. Or even epigen, which instructs cells in our body to make new red blood cells. These type of proteins have been developed for diseases that have enabled us to improve health of people and really improve the quality of and lives of so many people. And so at Juvena, we are leveraging our platform and technology to have enabled us to discover and now develop a fusion protein, which is the fusion of two naturally occurring human proteins that enhances muscle regeneration, enhances muscle metabolism, and by doing so, in our animal models to date, it has shown that it can reverse aspects of muscle wasting, improve strength and function, and overall improve metabolism. And we are so excited to be at the point now where in our development cycle, where we are gearing up to be ready to dose first healthy volunteers to assure safety before going into people living with myotonic dystrophy. So our healthy volunteer trials should start later this fall, actually. And the way that we're going to be administering our drug candidate is through subcutaneous administration, meaning right underneath the skin, similar to the way that insulin is administered for people with diabetes. We would not be here today in the development of JUV-161 if not for the engagement of so many incredibly talented individuals and groups, including the Myotonic Dystrophy Foundation, which we're incredibly grateful to and thank for their support, the various government funding agencies, including the California Institute of Regenerative Medicine and the National Institute of Health Small Business Innovation Research, several of our expert clinical advisors, our leadership and staff, and overall many investors in Juvena and multiple stakeholders who are just as passionate as we are in really bringing therapeutics to market that can ultimately restore function to rejuvenate lives. Importantly, I would like to quickly thank Juvena's leadership as well as all our staff who are ultimately the foundation and soul of the organization and working so hard each and every day to get our therapeutic to the people who need it the most to transform lives. And I'd also like to thank our many scientific and clinical advisors who have been instrumental in lending their industry veteran expertise and know-how in helping us to discover and to develop JUV-161 towards the clinic, including our, for example, John Day, who is going to speak next, who is our clinical advisor, and we're grateful to him and many others for their support and for helping us share with the community what's in store and what's coming up in our innovative approach to treating myotonic dystrophy. Thank you so much.
Great. Thanks, Dr. Yousef. If you want to go forward to the next slide. I'm here to talk about an aspect of myotonic dystrophy that we don't always talk about. And I'm excited to do that in its relationship to Juvena's approach to treating myotonic dystrophy that is different than many of the other companies involved. I like the foundation. I'm eager to see every company succeed. In terms of Juvena, that's taking a different approach. Now, this audience well knows the complexity of this disease. This is a family that I worked with in Minnesota before moving to Stanford, and everyone in this picture is affected by myotonic dystrophy, demonstrating the diversity of effects for an individual. Some are mildly affected, some more moderately, some more severely affected by various aspects of the disease. Next. And this audience also is well aware that myotonic dystrophy affects many, many aspects of the body, if not all of them. Next, the important one that we all oftentimes focus on is muscle. And that's partly because of its myriad effects and important effects on how people function. But it's also true that we understand muscle much better than we do other tissues. We can study it so deeply that we really understand the effects of myotonic dystrophy on muscle in a lot of ways. So if we go to the next slide, we can talk about what many of those features are. Now, myotonia is obviously mentioned in the name, myotonic dystrophy, so everybody is aware of that aspect of the disease. We would term that more of a physiologic aspect. It changes how muscle functions, but muscular dystrophy, which is also mentioned in the name, is a description of muscle structure. And it says that the muscle is actually falling apart and damaged and replaced by scar tissue. You can see that on the right, where certain cells are damaged and being removed. The white spaces oftentimes have increased scar tissue in them. That's indicative of the dystrophy. What we don't often talk about is the fact that in addition to those two elements, muscle often just doesn't work well. And that we would term a myopathy. So it's not that the muscle is damaged or being replaced, but it just doesn't function well. And that can be due to a myriad different causes, some of which I've listed there, including just an impaired development where certain muscle fibers on the right are very small, and that can be because they just don't fully develop. They don't turn into mature muscle fibers well, and consequently don't work well. Or there's impaired regeneration of the muscle, where the muscle tries to repair itself, but ends up with a lot of those nuclei in the center of the muscle, which is indicative of the fact that the muscle just isn't doing its repair mechanisms right. Or there's alteration in the contractile force. It just doesn't develop strength the way it should. So all of those are elements of myotonic dystrophy. Importantly, there's also an inadequate energy production, so that there can be an absence of certain genetic products that are really critical in terms of energy production or metabolism within the muscle fibers. So how does all of this happen from a disorder that affects one gene? You get one gene and all of these effects. And Dr. Rohrwasser already alluded to this. And if you go to the next slide, we can see many of you are well aware that myotonic dystrophy type 1 is caused by this CTG expansion. And next, myotonic dystrophy type 2 is caused by a tetranucleotide CCTG expansion that, if you click ahead one more, you can see that both of those cause those clumps of RNA that Dr. Rohrwasser was referring to that alter the whole mechanism within the cell. So the family of genes are misspliced or misprocessed within the muscle. And so this RNA accumulates and causes that effect on all of these different aspects of muscle function that we just alluded to. For instance, if you go to the next slide, we can focus on the insulin receptor. So muscle is an important tissue for insulin activity. And if you look at the normal muscle there, you can see that in normal muscle, 2/3 of the insulin receptors are created in a way that they're very responsive to insulin. And even in other diseases of muscle, wherein other than myotonic dystrophy, that percent of insulin receptors that are responsive to insulin stays above 50% there. But in both myotonic dystrophy type 1 and myotonic dystrophy type 2, that ends up being switched so that the percentage of insulin receptors that are doing their job and responding to insulin goes way down, goes down to 29% in that instance or 19%. Furthermore, on the next slide, if we look at the muscle of an individual who was biopsied twice, 8 years apart, on the top, we can see that her muscle looked in that pink box. That just looks perfectly normal. That is a very normal appearing muscle. But you can already see, even at that point, she had those pink clumps in the nuclei of those fibers, and they were already affecting the insulin receptor. So rather than being 67%, which it should have been if it was completely normal muscle, it's already down to 26%. And if you biopsy her a second time, 8 years later, that the muscle now looks much more altered by the myotonic dystrophy, and that insulin receptor activity has fallen down to 4% rather than 67% being normal. So I think you get an appreciation of the impact of this kind of thing on muscle. So this leads to these conclusions in the final slide, where sure, there are treatments that we are very optimistic can slow down progression of the muscular dystrophy, which we are very optimistic about. But there are also treatments that we can hope will improve the muscle function by focus on what I've termed those myopathic features, the features of muscle that are preventing it from functioning normally. And those features can be due to a specific genetic change. You can think of mexillatine, which many of you have heard about, which alters the function of a single gene product to improve the myotonia in some people. So we can focus treatments on single genetic changes in myotonic dystrophy, potentially with really impactful treatments that we're excited to focus on. So I think there are important movements going forward here, and I'll just put in a final plug. I don't know if this got on the final slide if you click forward, but it's that I really want people to focus on signing up for research. We need more myotonic dystrophy type 1 and type 2 people involved in research. If you can reach out to the MDF, we're excited to move that forward. So I'm going to pass off to Jeremy, but thank you very much.
Thank you, Dr. Day, for that great overview on the research from you and colleagues establishing aspects of the molecular basis for myopathy in the progression of myotonic dystrophy. Next. As Dr. Rohrwasser mentioned, Juvena is developing a drug targeting cellular dysfunction, aspects of the disease we believe could address myopathy. But what is cellular dysfunction? And to put a picture to that word, next, we really need to put those 4 steps of DM progression where they live in context, which is cells. And so you can see here is an example of a human cell, which has an inner compartment where the DNA and the RNA typically exist, and then a middle compartment where most proteins exist, like the ones that make your muscles contract, and then an outer layer where cells send and receive signals with proteins called receptors. Receptors are like antennae, receiving signals from the outside of the cell and transmitting them into a message for the cell to respond and do something. Next. Some of these receptors don't function properly in DM, sometimes because cells manufacture the wrong version, such as the case of the insulin receptor Dr. Day just highlighted to us, and sometimes because the internal wiring of the cell is in the wrong state from responding to the stress response of the toxic RNA. The family of insulin receptors is particularly important for muscle, where it drives sugar uptake and the push for cells to grow and survive to achieve proper muscle health. And when the signals that come from the outside of those receptors are weak, the muscle cells don't maintain those contractile parts of the cell or take up the sugar they need for the energy to drive proper muscle function. And they can become weak, which was the myopathy progression Dr. Day gave us a great example of. And that's an example really of cellular dysfunction in action. There are potential solutions to this. Next. At Juvena, we are advancing our drug, Juv161, a signaling protein we engineered to boost signal through the IGF family of receptors. It's comprised of two main parts. Next. On one side, there's the insulin-like growth factor 2. This is a member of the insulin family that we've engineered to be able to boost the signal through some of these pathways. It normally plays a role in driving the generation of muscle during the earliest stages of development, but the receptors, antenna, it hits are still present and active on the surface of adult cells, even if sometimes in altered forms. Juv161 can hit even the altered version of those antennae. Next, we've fused it to another protein to improve how long it lasts and how well it can reach muscle, protein called human serum albumin, highlighted here in gray. This is a naturally occurring protein, one of the most common proteins in human blood, and it does this function well. Next. Juv161 can activate the misspliced version of the insulin receptor and other members of the insulin family to boost signal through this critical pathway to improve many aspects of muscle cell health and potentially improve muscle function. Next. Some of the very quick overview of some of the data that gives us this conviction from mouse models where we see improvements in muscle mass at the muscle group level, in part driven by an increase in the muscle fiber size, which is the graph on the right. Next slide. Importantly, these fibers are also able to respond to glucose better, which is the fiber typing you see on the left and in the middle graph for blood glucose levels in the middle. And these improvements in muscle mass and muscle metabolism not only halt the loss of muscle strength we see in this model, which is the vehicle-treated black line on the far right graph, but can improve it over time when treated with Juv161, which is the green line on the grip strength graph on the right. These data give us a lot of conviction that Juv161 is well positioned to address myopathy in myotonic dystrophy. And with that, I'll turn it over to Dr. King to describe our plans to translate this work into the clinic.
Thanks, Jeremy. Sorry for the technical misfunction there. Based on the information you just heard, what we believe is Juv161 is going to restore normal function to muscle cells. And it does that partly at a cellular level by increasing muscle cell function, muscle fiber structure, and then ultimately muscle strength and endurance. And that translates at a personal level for patients to increases in mobility, increases in social participation, and quality of life. Next slide. So as a result, we're now in the process of moving Juv161 into the clinic. Our approach so far is that we're going to start with healthy volunteers. And what we're doing with that part is to look, first of all, at a single dose escalation study. So we increase the dose of Juv161 in this group of healthy individuals. We're looking primarily for safety, make sure that the drug is safe as we move forward. And then from there, we begin to administer the doses in multiple occasions so that, again, we can demonstrate safety for that group of individuals as we move forward. As a critical part of that, we also measure how much of it gets into the blood, and we'll try to assess some certain markers that might be associated with its efficacy or benefit in patients. From there, we will then move into a group of individuals who have myotonic dystrophy. We'll do a couple different types of studies. First, to prove that the drug actually works in that circumstance and improves muscle function. And they'll do a larger study to demonstrate more convincingly, I suppose, from a statistical standpoint and from a functional standpoint, that people do in fact benefit from that. One of the great aspects of this particular compound is because it acts at a muscle level, it has the ability to potentially work in multiple other indications, not just DM1 alone, but maybe in DM2 and certain other muscle disorders. We won't know that quite yet, but we, as Dr. O'Connell just mentioned, we have a strong conviction based on the information that we've looked at so far that, in fact, this will happen. Next slide. So it's a sort of a very high-level overview of what we're planning to do. We'll first of all do this phase 1 study, single, multiple doses in healthy subjects. From there, we'll then move into patients who have the clinical diagnosis. We'll be looking for genetic confirmation to make sure that we're looking at the correct type of individual. And then, as I mentioned, we move into this proof of concept study. Then we'll move into pivotal studies. And what we will also be doing is that once individuals complete these particular trials, they'll then move into what we'll call open label extensions, which means that if you as a patient have benefit from this drug, you'll have the opportunity to remain on it up until a point in time where regulatory agencies approve it for actual sale at some other point in time. Next slide. So what we'll be looking at, the kinds of things that we'll be looking at in our studies, are to measure things that are, we believe, beneficial to patients. So on one level, we're looking at things that reflect your ability to move, walk, run test, how you can get up out of a chair, and then being able to move from that standpoint. Also, we'll be looking at the myotonia. Our belief is that this will be a secondary benefit from the drug, not necessarily the primary, but we'll also be looking at that. We'll also look at strength, and we believe that that will be an effect of the drug following its administration. We also know that, and Dr. Day pointed this out earlier, that the disorder has, as you know, many other manifestations. We'll be looking at some of those effects in our studies. We're not quite sure at this point whether or not the drug will, in fact, impact all of those, but we'll certainly look at them, and clearly, if they do, then that presents additional benefit for you as the individual patient. One thing that's of importance, of course, is to look at muscle mass. We'll be looking at the function, we'll look at strength, we'll also look at muscle mass, and then we'll be looking at various forms of assessments of your ability to move better, to do more things. And those will be the activity and outcome assessments that we'll be looking at. And we'll be looking at other aspects to it. And as I mentioned, we'll continue to look at the pharmacokinetics so that we make sure that the blood levels that are present in individuals after they've taken the drug correspond to the effects that we're looking at. Next slide. And so clearly an important question that everyone's going to ask is, well, when are we going to be able to see this drug or use it and participate in clinical trials? This is a really high-level overview of what that will look like. You'll see on the slide, it says phase 1. So that's that part where we're looking at the study in healthy volunteers. From there, what we call phase 2 is the one that corresponds to that next level study in patients. And then from there, we move into the phase 3, the larger study. And as I mentioned, we will continue to employ these extension studies so that if you have benefit, you'll be able to continue to participate. So the key for you, as Dr. Day mentioned, in terms of making sure that you register for participation through the MDF, The way you find out about our study will be to stay in touch with clinicaltrials.gov. That's where all of this information ultimately will show up, and that's how you'll find out about the trial. You can also, of course, find out about the company by looking on the websites, but for the clinical trial itself, the place to look is clinicaltrials.gov. And from there, I will hand back, and we'll say thank you to everyone for listening, and we're happy to answer questions.
Great. Excellent, excellent presentation. So just as a quick reminder, right, many of you have done, you have an opportunity to ask questions, put them in the chat. You can do that anonymously. And now I'm looking forward to a robust discussion and conversation. And we do this kind of as we usually do, right? We have our cameras on, and all present. So what I've been doing is I compiled questions you all submitted either when you registered or now during the presentation, and we obviously want to answer as many as we can. So the first thing I want to echo what Barney and John, Dr. Day just said. Please utilize, participate in the registry. We need your participation. There are many exciting treatments coming and trials coming down the pipeline, and a critical component is that you are registered, and that we can conduct clinical trials. Another housekeeping issue, of course, we make this presentation available as I stated in my introduction. We will try. We will have it available probably no later than early next week. Okay, so with this, if we could now get the Juvena team back on. So kind of my groupings are, if you could quickly repeat the dates when you anticipate starting the trials in healthy volunteers. Quickly state again the method of delivery and the frequencies, and talk about the inclusion and or high-level exclusion criteria for the trial.
Sure. In terms of the healthy volunteer studies, those will begin later this year. They'll last until probably the beginning of the second quarter of next year. And then shortly thereafter, we have to do regulatory filings, making sure that the regulatory agencies are comfortable with the information that we've generated in those particular studies. And then from there, we'll move into the clinical trials in patients. So again, your notice of that is that there'll be an announcement on clinicaltrials.gov, so you can find the information there. With regard to the drug itself, the drug will be administered via subcutaneous administration. That means that there will be a small pinprick in your skin. The drug will be delivered that way, and then we'll look at it over a period of time. For patients, the expectation is that we'll be administering the drug once per week for the period of time that we're looking at the studies. The entry criteria, I mentioned that you need a diagnosis and you need confirmation from a genetic standpoint that you do have myotonic dystrophy type 1. There are some other criteria that relate to your ability to move, not having certain other diseases and so forth. But those would be fairly standard. And if you know of other trials that are being done in the area, our criteria for inclusion and exclusion are very similar to what those studies look like at this point in time.
Excellent. So two more questions in the same categories. If you could explain whether the drug will need to be taken regularly, like insulin, and how do you see that actually, assuming you get approval for the drug, how do you think how frequently would dosing need to occur?
Sure. Yeah. So our plan is, as I mentioned, the drug be administered subcutaneously. In the initial portions of the trial, that dosing would happen at the doctor's office. So the site where you participate in the clinical trial, you would receive the drug from them at that point in time. Once we get to a little bit longer studies, so now we get into that 3-month kind of study or 6-month kind of study, the first couple of administrations would be, again, done at the site, but we would teach you how to do it. And so that from that point forward, you would be administering the drug yourself, as I said, into the skin. So our plan is that we'll have the drug in a formulation so that it'll come in a syringe, and then you can inject this drug into the skin yourself as we move forward with the drug.
Great. So now let's go a little bit towards mechanistics of the drug. And for this question, I would like to put you all on the spot a little bit, including Dr. Day. So assuming the drug is effective, where would you, in your respective crystal balls, think we would see effects first? What would what would your greatest hopes in terms of improving symptoms, right? What should we see? What could we see first? I don't know. That's a difficult question. Maybe we can, if you can all take a turn, that would be great.
Sure. Let me start. I mentioned that we're going to be looking at several different things during the course of the study. What we would expect first is that we would see changes in muscle strength, and I'm using my hand, but our expectation would be that would apply to legs and so forth as well. And then what you would notice as a patient would be you'd have a greater ability to do things with your hands, and you'd be able to walk more efficiently and safely, I guess I will say. What we don't know beyond that is how the other effects will happen over a period of time, but at least that would be the primary initial aspect that we would anticipate. Dr. Day?
Yeah, I think it's a great question. I think it comes back to this varied kind of impact of myotonic dystrophy on muscle. And, we can think that muscle that is very damaged by muscular dystrophy is unlikely to respond to this type of treatment. We're going to be looking for muscle that is not functioning well, but is still intact. And so that's going to differ a lot from person to person. But we can think that we could see improvements in strength, but also, importantly, maybe improvements in stamina, so that people will have improved energy levels and ability to continue to do activities for a longer period of time. But I think we will see differences from person to person. And that's going to be some of the exciting work that will come out of the initial trials.
Okay, we also want to hear from you, Dr. Yousef, and then from Dr. O'Connell.
I think I would just like to echo our Chief Medical Officer, Barney, and Dr. John Day, where I think that what will happen the soonest will be really at the cellular level, seeing these improvements in muscle, anything from the circumference of our muscle to its metabolism, its ability to really respond to insulin sensitivity, and eventually, with some time, and hopefully not too much time, but if you consistently take this therapeutic, you will then really start to see those improvements in your mobility, in your energy levels, in your ability to really live your daily life with a bit more energy and mobility than hopefully prior to taking this, and to hopefully keep maintaining that over time to really slow down the progression of the disease would be the ultimate goal. And not just slow down, but ultimately build back your muscle and help repair it. So that's our goal.
Jeremy?
I'm not sure how much there is to add after all those excellent answers, but I guess the one point I might comment on is in adults, IGF-2 is made in small amounts following exercise to improve recovery. And so it might not only improve aspects of muscle when being used, but the ability to recover better afterwards. And so perhaps improved exercise tolerance, if people are up for it, could be one of the potential benefits I would hope for.
Fabulous. So the next question is, I would like to propose we do a similar go around, but here we start with the physician and advisor. So the question is, would this work? We always talk about DM1, right? But here we might also have a candidate that might be that might work for DM2. So the question is, would this, of course, work for DM2, and why the focus on DM1 first versus DM2? What was the rationale? And I think this would be really good for our community. Maybe let's start with Barney and then John.
Sure. Let me start with first of all the why of the DM1 first. When you do clinical trials, one of the challenges is to identify a group of individuals whom you think will benefit from the particular therapy you're going to use. And while today we think that ultimately this drug will have benefit across several different muscle diseases, we have to start in one place because we have to demonstrate that it works there, and then we can move across to other studies. So that's the rationale for DM1, and the rationale for DM1 versus DM2 is patients who have DM1, there's a very broad range of the manifestations of the symptoms, as Dr. Day showed in the one particular family with their disparate presentations. But we're trying to identify a group of individuals who are a little more severe, so hence DM1 versus DM2. Again, we think it will work in DM2, but we've started with DM1 as the initial indication. John?
Yeah, I'm so glad you brought this up, Andy, because I very much wanted to make this point as well. Because I know how much the DM2 community is eager to get into clinical trials. And I do think that this is something that can definitely impact both forms of myotonic dystrophy. For me, part of the reason, as Barney just alluded to, starting with DM1 is because we can get the trial done more quickly. Unfortunately, we don't have as robust a registry yet for DM2. And I think anything we can do to encourage the community to sign up is going to go a long way towards us being able to get some of these trials done in DM2. But we're quite optimistic that a treatment like this that works in DM1 should work in DM2. So if we can prove it in DM1, there would be a smooth glide path to us showing that this works in DM2 as well.
Great. Dr. Yousef and Dr. O'Connell, do you want to add anything, or do you was that enough?
I think it's great. Maybe just adding that we're actually what we're the most excited about is that in many ways this therapeutic we're developing is like the insulin of muscle, and we have in fact seen that it works towards restoring even aged muscle function from aged animals through to different types of dystrophies. So we are really hopeful and really want to get this out to hopefully treat many types of muscle diseases and really ultimately rejuvenate the lives of millions of people worldwide. And we're so grateful for this opportunity to be at this point and hopefully get our drug to market soon.
Great. Okay, so another somewhat related question: the congenital form of DM1. So, sorry, Barney and John, I put you on the spot again first as the medical experts. Can you what are your thoughts there? Will the drug be a candidate for these subtypes as well, and how soon? And the floor is yours.
Yeah, I get stuck with the how soon part. I think the answer is yes. You will have applicability. As you do drug development, we mentioned that we'll start with the M1, then we'll have the ability to expand beyond that. At a point in time where we have evidence that in people who have the disorder, DM1, that we have clinical benefit, what that does is then open up a really broad range of potential opportunities. And I actually have John talk more about the congenital version and other manifestations, but that would be our plan, would be that ultimately we would expand as much and as far as we can.
Yeah, I would just add a little to that, and that is that we know that in the congenital cases that there really isn't much dystrophic change, as I was showing early in the slides, and consequently that muscle is just not functioning right. It really has much more of a myopathy than a dystrophy, and so I would be very optimistic that anything that can really conquer or affect and treat the myopathic features of this disease are going to have a significant benefit in that congenital group. So I'm excited about that trial where we can look, because it's almost a pure form of myopathy as opposed to dystrophy, and we can look for significant effects there.
So now I want to shift a little bit towards other mechanisms of the disease, and if you could expand a little bit on penetrance of the blood-brain barrier and targeting, but the potential, right, of this intervention targeting other clinical presentations, especially CNS presentations.
Well, so far, and Jeremy can spend more time talking about the specifics of this, we don't believe that the drug will get intact anyway into the brain. So it does not pass the blood-brain barrier. What we, again, clinically hope, we won't know until we do the studies and look at the data. We will hope that there will be other effects, such as particular things like sleepiness or fatigue. Those would be things that we think will impact ultimately. Some of the other things, such as changing cognition, we just don't know from a clinical standpoint about those at this point in time. Jeremy, other thoughts?
Just to confirm, yeah, based on the structure and sequence, the intact molecule is not believed to cross the blood-brain barrier at present. We'll gather more data on that in our early clinical trials.
The physicians, when let's again assume, right, the drug is the drug is effective. How would you monitor and stratify and recommend use, right, for for patients with different types of symptomology? And do you think, can you comment on the potential of this drug as a combination therapy with other drugs, right? That might target other mechanisms.
Well, yeah, I'll start. I think from the standpoint of the monitoring therapy, we believe, and again, the caveat is always we have to do the clinical trials to generate the information for sure. We believe the drug will be safe, otherwise we wouldn't really progress it into people. But from the monitoring standpoint, we'll always be looking at potential side effects, but from the benefit to an individual, the expectation would be that there would be some improvement over time that would accumulate. And so there would be progression in the benefit across time. From the potential side effects, we talked about this insulin pathway. So the only thing that we think is a potential real issue at this point in time is that the blood sugar could be affected. So that would be the one thing that we would monitor. Now, that being said, by administering it subcutaneously, so again, in the skin, so the distribution of it is not immediate, but goes out over a period of time. And we're asking people to take it after they've already had food. We believe that particular side effect will be less common or occur less frequently. So from the monitoring standpoint, that would be our plan long term. With regard to speculation about potential combinations, we don't see a reason why the drug couldn't be used with, for example, the drugs that are trying to attack splicing abnormalities. A priori, there's no specific reason that it wouldn't. And the expectation is that it would, in fact, be complementary to those particular medications. So with that, I'll turn it back to John for his thoughts as well.
I was going to turn it to Dr. O'Connell for his thoughts. I don't have anything really to add, I don't think.
Yeah, just to echo, I think we don't see any reason it wouldn't be additive with therapeutics targeting steps outside of the cellular dysregulation, or perhaps even therapeutics targeting other aspects of cellular dysregulation. As noted, it should be additive with things that target, say, the myopathy, the myotonia aspect, which is not an aspect we would directly touch on with the mechanism of Juvena 6.1.
Right. So let's wrap it up with two more questions. So the one is very specific. The other one is more broad. Let's start with the broad one. Do you think the drugs will reverse symptoms or stop progression?
Well, again, speculation. My perception is that if you look at the drugs that are directed toward the splicing abnormalities, one of the important aspects of that would be stopping progression, so reversing those cellular defects that are contributing to the manifestations of the disease. What we don't know at this point is how much they would restore function versus preventing further decrease in function. If we combine the drugs, and keep in mind there are a lot of ifs here, and we won't know till we actually generate the data, possibility would be a scenario that would say those drugs stop the progression of the disease. There's some improvement in the symptoms that goes along with them. And then you add in a drug like JUV161, and now you're improving the muscle beyond what you might have expected with the other drugs. So the combination, if you think about it as a potential combination, that would be one possibility about how it might function across a period of time. John, thoughts?
Dr. Yousef, I think maybe had another thought I'll ask.
Yeah. Just my only kind of, quick just adding to that is, based on our preclinical research in animals that are modeling the disease and the disease progression of myotonic dystrophy, where they have the CTG DP kinase gene mutation, while the control animals are continuing to decline in muscle function, the animals that are getting Juve161, not only is it preventing the progression, it is actually building back strength and function based on our readouts. So there is a chance, although we won't know until we really test this clinically, but there is a good chance based on the mechanism of action of the protein and its ability to restore and repair muscle function, that it could improve function with time, especially if that's combined with things like exercise, good nutrition. And so we can't wait to test that and see that, and really are hopeful that that might be the case.
Maybe I'll just add one point more general to that, Andy, and that is that, I think we all hope that one of these treatments that are in trials is just going to be a grand slam walk-off home run, as I often say, and end myotonic dystrophy as we know it. That's not a common experience in drug development. We usually take a bite out of the condition, and we have more to do. And I think at this juncture, it's really important for us to keep moving on all fronts, because I think combination treatments might very well be very meaningful and very impactful as we move forward.
Great. So the very last question is a very science and very geeky question, whether you could speculate on the differential effects of IGF-2 versus other factors such as anti-myosin agents. And after that, we wrap it up. And I would like to conclude and be respectful of everyone's time. I think maybe this should go to the scientists, right?
I'll take a first stab at that. Assuming that was for an anti-myostatin agent as the comparator, I would say they're different. And if pardon this more scientific answer to a more scientific question, the receptors activate very different pathways for modulating how cells maintain or build muscle function. The myostatin pathway really is about maintaining the number or amount of contractile fibers within a muscle cell, whereas the IGF family has more to do with improving glucose uptake, energy utilization within the cells, has some regulation of the mitochondrial function, as well as the survival and downregulation of some of the stress response pathways. So they do converge through aspects, but there's really a difference in those two pathways, particularly during new muscle cell formation, where IGF-2 can drive the survival and differentiation of the muscle stem cells to be adding into the muscle groups, as opposed to the myostatins, which really act more on the existing muscle fibers themselves. And I will, of course, defer to Dr. Day for any correction to that point.
No, I have none. I again, I think both could have an effect here. But I think, and the answer is going to be in the clinical trials, and we'll see what happens. But I think there's every reason to think that we're approaching this. I mean, there's been a long history of people trying to develop IGF-1, kind of agonists, and I think approaching this the way that Juvena has, with this prolonged slow distribution of IGF-2 agonism, is very exciting and could be quite impactful. So I'm excited to see what we see.
Right. With that, let's go to our very last slide, because I want to make a few comments. So if today's program was interesting, right, and it did speak to you, please consider making a donation to MDF. If you are living with the disease, please participate in myotonic dystrophy. I want to iterate this again and again and again. We need your participation. Clinical trials can only happen with well-characterized patient cohorts, and clinical trials cannot happen without you. There were some questions with regards to the mechanics and the mechanistics of clinical trials. You heard, and there are updates. There will be regular updates to clinicaltrials.gov. And as we learn from Juvena, with whom we will maintain and have been maintaining regular relationships, that there are trials in other countries and other sites, we will, of course, notify the community. So only together can we change, right, this disease. And with this, I would like to end. I would like to thank the Juvena team for a very nice, very good presentation and the very, very robust Q&A session. We will have the video available as probably no later than sometime next week. And with that, thank you again. Thank you very much for participating, and we are looking forward to seeing you again for the next event. Thank you.